Good morning and welcome to today's MPG Primer. We are very fortunate to have two speakers for today's session. The first speaker will be Dr. Joshua Pan, who is a postdoctoral fellow with William Hahn at Dana-Farber Cancer Institute and at Broad from 2018 to 22. He collaborated with Marinka Zitnik as well as Cancer Data Science and DepMap on this project. His current position is as a research scientist in genomics at DeepMind in London in the UK. Our second speaker today will be Dr. Marinka Zitnik, who is an assistant professor at Harvard with appointments in the Department of Biomedical Informatics at Broad Institute and at Harvard Data Science. Dr. Zitnik investigates graph representation learning, including pre-trained, self-supervised, multi-purpose, and multimodal models trained on broad data at scale. Dr. Zitnik has published extensively in top machine learning venues and leading scientific journals. Her research won Best Paper and Research Awards from the International Society for Computational Biology, the Barer Early Excellence in Science Award, Amazon Faculty Research Award, Roche Alliance with Distinguished Scientists Award, Rising Star Award, and the Next Generation in Biomedicine Recognition, being the only scientist who received such recognition in both categories. Again, thank you to both of our speakers for joining in today. And Josh, please get started whenever you're ready.
Thank you, Diane, for the wonderful introduction. So the structure of today's presentation, the first 30 minutes or so, I'll be presenting some collaborative work performed with Marinka, who's on the call, as well as William Hahn and James McFarland, all at the Broad Institute. And then we'll zoom out a little bit, and Marinka will present about representation learning at large. So the theme of today's presentation is representation learning in biomedicine. And so hopefully you get two different views on that topic today. Feel free to address your questions to myself or Marinka through the chat, and we'll do our best to answer. So the structure of this particular module in today's presentation, I'll introduce sort of a conceptual correspondence between words and genes that I think helps introduce the concept of pleiotropy. Then we'll talk about a representation learning method called Webster that infers pleiotropy from certain kinds of biomedical data. And then we'll open up a few discussion points sort of connecting to themes that are in the literature at large. But let's start with our introduction today. So one of the ideas I want you to hold on to as you go through this talk is that words and genes share a correspondence, which is that their meanings arise from their context. And so to give an example of this for words, apple is what's known as a polysemic word. So Google understands this. If you're searching for apple in a particular context, it'll return results relevant to that particular context, depending on what the word apple is referring to. And so the sentence context sort of resolves the particular meaning that a polysemic word can have. So at the high level, genes or protein products that arise from these genes also have this property. So for example, H2AFX is a pleiotropic gene. And so a pleiotropic gene produces a protein in this case that has the potential to activate different kinds of DNA damage response pathways, depending on the kind of chemical insult that's being delivered to, let's say, a cancer cell. And so in this case, different toxins can produce double-stranded breaks. H2AFX can respond and sense those breaks, but they can activate different pathways as repair mechanisms. So once again, in this case, the cellular context induces a particular function that is potential within that gene or protein. So to sort of connect this to a wider theme in the representation learning, let's say a field of or machine learning at large, how do we approach these kinds of concepts in those fields? And so it turns out that unsupervised learning of word polysemy is very common. So the general approach here is that you can start with a corpus of sentence contexts, let's say like a Wikipedia, for example, and you can train a model called a word embedding that takes as input that data, and it outputs a vector representation of each word in your corpus. So now a word is represented as a series of numbers. And what happens in this method is that words now have linear semantics, which means you can manipulate them in a particular way. So for example, a famous sort of example in this field is if you take the word embedding of the word king, subtract the word embedding for the word man, add the word embedding for the word woman, you'll approximately recover the embedding for queen. And so these kinds of linear relationships are intuitive to us at the high level, but the machine learning models can capture that in this sort of latent space representation that it learns automatically from the sentence corpuses. So that's actually quite common and well understood. But unsupervised learning of the analogy in genetics, this gene pleiotropy, is relatively unsolved. So, it's still an open question, for a gene that has multiple potential functions, what kind of data and what kind of approach should we use to kind of recover these linear semantics that we sort of suppose that genes might have? For example, a pleiotropic gene can have multiple functions, et cetera, et cetera. So a group of collaborators, including myself and Marinka, really wanted to approach this problem. And that's the goal of today's talk, is to walk you through what we've discovered. And so if you're interested in exploring more, this paper has been published, and there's actually a mini site on the depmap.org portal describing this method. But I'll walk us through sort of how it works and give you a few examples on some real data derived from human self-fitness screens. So as I said, we had to start with something. What kind of data should we use? And there's tons of different kinds of biomedical data that we can think about starting with. For biological functions, I think a powerful starting point are these gene perturbation effect measurements. So the Broad Institute has actually produced a lot of this kind of data. The general strategy here is to start with the cell context, like a cell line, and introduce, let's say, CRISPR guide RNAs as a perturbation vehicle. And this results in a group of cells that have a particular gene, one in each cell, knocked out. And then you can apply sort of a selection pressure, maybe just watch those cells grow over time, and you measure the fitness consequences of that individual gene knockout. And maybe you have multiple measurements because of the way you designed your assay. But at the end of the day, what this boils down to is, for your particular cell context, you've knocked out a panel of genes and you observe some outcome. Let's say changes in fitness growth rate. So what's really cool about this method is that you can scale it across different chemical or sort of genetic combinations to produce different kinds of cellular contexts. And there are many different kinds of examples of this data set, of these kinds of data sets now. And these are really exciting because they give kind of a causal indication of what that gene might be doing, right? Because you made measurements without and with the gene. So you have that direct comparison for some particular fitness phenotype, let's say in this case. So let's start with that data, and then what kind of approach might we use? So this is very much inspired by some of these word embedding approaches that I described earlier. But it actually comes, there's a sort of principled approach to this that was described in 2011 by Gunther Wagner and Jiangqi Zhang, where they wrote about the concept of pleiotropy and how you can think about pleiotropy in sort of high dimensional phenotypic space. And they outlined sort of 3 criteria back in 2011 that we should follow if we're thinking about pleiotropy. One is that genes and functions tend to have sparse connections. So a gene might have a particular set of pathways that it interacts with, but maybe not too many. If we're thinking about high dimensional data, like these fitness phenotypes across cell contexts, and we're thinking about, okay, maybe there's a latent variable that we're looking for in that space, those latents have to be biologically meaningful. Otherwise, your calculations may be off about how many functions your gene might have, right? And finally, they are very clear about the fact that different cell contexts might have redundancies. So if cells share a tissue of origin, for example, that are similar, they might sort of respond to the same gene knockouts because they have similar pathways that they depend on for growth. So our approach, we nicknamed it Webster. And it kind of follows this roadmap that I've outlined, where we start with a panel of fitness data. In this case, you have a set of genes knocked out over a set of cell contexts. And we'll see a few examples of that next. But that's your starting point. The approach itself, it's actually called dictionary learning or sparse approximation. You can also think of it as matrix factorization or compression. There's lots of different ways to think about these classes of methods. But it really just boils down to learning 2 matrices from your input matrix. So the first matrix in red here has dimensions m by k. So that's the number of cell contexts by a number of functions or latent variables. And that k is a number you can choose, but it's a low rank. So k is small. So you're trying to learn a set of k functions from a set of n gene perturbations. Okay, so that's kind of your first output. The second output is kind of, you want to map your genes to those functions, right? So that's kind of the other part of this goal is, let's say my gene probably has 2 or 3 functions. That's captured by a parameter named T, or called T here. And the key is that each gene is mapped to a sparse set of these K functions. And the way that you do this is that this matrix multiplication has to approximate the input data as best as possible. So it has to fit the original data while obeying these sparse approximation constraints. So this is actually a very classical setup in machine learning, and many methods can be broken down into this low rank factorization task. How can we incorporate a few biological priors to make this slightly more meaningful or specialized for the kinds of data that we are interested in analyzing? So the first thing that we do is that we regularize. And so what this means is, for those of you who are in the literature about CRISPR coessentiality, screens and such like that, you might know this already. But columns in these sort of data sets, right? So these gene perturbation effect columns, these measurements have a very stereotypical correlation structure. So for example, let's say these 3 red genes are all part of the same protein complex. Well, knocking them out across a set of cells might induce very similar fitness phenotypes. So similar, in fact, that you can take those profiles and sort of cluster them, and you might get clusters that look like your favorite protein complexes. So this is a well-known result in the CRISPR screening field. And what you can think of is all of the columns in this data have relationships in those correlations that you can think of as a graph. And so this co-fitness graph is something that is very useful to keep in mind. Because when we map genes back to functions, we want to make sure that genes that are very correlated in the original data, meaning that they might be part of the same pathway or something, are being mapped to the same functions. And the closer we are doing to sort of following this graph, the better we might be doing in capturing realistic or biologically meaningful latent variables or functions as we're doing this compression. Another thing, and again, I mentioned this in that sort of review from 2011, but cell contexts share redundancies. So for example, if you're screening several cell lines from the same tissue, those tissues may harbor the same genetic dependencies. And so those relationships between cell contexts also need to be captured in our dictionary matrix in this case. So in our functional representation of the data, similar cell lines from the input should share similar inferred functions that they depend on. So based on those rules, we have this approach at the high level. This is a bit technical, so I'll walk you through the high level here. But basically, to run this approach, you start with a matrix of fitness effects, as I described. You produce these low rank approximations here, such that D and X multiplied together approximate Y. And then you do your best as possible to obey these correlation constraints across rows and columns in your original data. And when you're performing this task, there are a few hyperparameters that you are very careful about tuning. One is K, which is the number of functions you're trying to learn, and T, which is the degree of sparsity that you're assuming exists in your system. But at the end of the day, this boils down to sort of learning these latent dimensions or variables in your data and mapping gene effects to those. Okay, so that's a whirlwind tour of the method itself. I'll spend the next 10 minutes or so talking about examples and what the impact is of running this kind of analysis on, let's say, your favorite fitness screening data set. So I'll discuss 2 fitness screens that we applied our method on. This one is from the Durocher lab in Toronto, Olivieri et al. And they performed a very cool screen. So what they did was they started with a parental cell line, which is called RPE1, and they applied 31 genotoxic treatments to those to that exact parental cell line. So each of the rows in this matrix here, in this treatments, these are that parental cell line treated with some drug, or in this case, UV light. And so the whole point of that is to generate a set of cell contexts that you can then perform geno-wide screens over. So the way that this data set works is, let's say I knocked out some gene, and in the presence of some drug, cells die when you knock out that gene. And when there's no drug, the cells are doing fine. And what that might mean is that, okay, when there's this specific genotoxic stress induced by my chemical, that gene is responsible for repairing it and sort of recovering this, recovering the cell state. So it's really mapping, genotoxins to DNA damage response genes. So as I mentioned earlier, so our approach attempts to compress this data or learn meaningful representations of this data that allow for sparse modeling of gene effects. And so this is the input data to their from their paper that we downloaded. And as you can see in purple, there are some genes that are essential in the presence of some treatment. And so those are some of the sort of modules or factors or clusters that we want to hold on to and learn automatically in our representation of this data set. And so this is the result of applying our approach. And so you can see that in our reconstruction of this original data, we've largely, let's say, captured these broad clusters or effects that we see like even visually in the original data. And so what's the difference between these two? So I'll show you. But basically, we've reconstructed that data in terms of 10 functions that we've learned automatically. And we've mapped each gene effect to 2 of those functions. So how does that work? So as I advertised in the model walkthrough, there are 2 key outputs to keep in mind. The first is this dictionary matrix. So this dictionary matrix here has the same number of treatments in rows as the original data. So there's 31 sort of genotoxins here. But now there's only 10 sort of functions. Okay, so you can think of these as latent variables or factors. I'm going to call them functions. You'll see why in a second. But each of these captures a specific or reoccurring fitness pattern that's present in the original data. The second output, as I mentioned earlier as well, is this loadings matrix. So each column here is one of your genes. There are about 300 or so sort of significant fitness genes in this data. So there are 300 columns here. But you can see there are 10 rows. And these are the exact same as the columns over here. And these are just these latent variables or functions that we've learned. And so the blue indicates that when you knock out that gene, it seems to be hitting this function and that function because we set this parameter t to be 2. So each gene gets mapped to 2 of those functions. So that's what the method is doing, okay? But now how do we interpret it? So what I've done now is I've put back in some of these annotations. So for example, these are the names of the treatments or the drugs that were used to sort of perturb these cell lines, this cell line. And then these are the names of some of the pathways that we've learned automatically from the data. And the way that we annotated these was we actually just sort of looked and sort of performed Go term analysis of which genes were being mapped to these latent variables or functions. So in this case, the first one that we learned here, number 1, it is enriched and actually recalls almost all of the canonical nucleotide excision repair genes, which you can see down here. These are literature annotations that we've sort of held out during model training and used to just kind of annotate the model outputs. So you can see that there's a large overlap between this learned representation of nucleotide excision repair that we got from our method and the canonical nucleotide excision repair genes. If you look over back in the dictionary matrix, here we have an annotation that UV induces a dependency on nucleotide excision repair, this sort of number 1 over here, which is exactly what we know from the biology. So what are the consequences for modeling individual genes? So as I mentioned before, what our model's trying to do is approximate these sorts of profiles. So for example, H2AFX was perturbed in the presence of all of these treatments. That's part of their screen. And this is their actual screening data. Purple means it's more dependent. So our approximation of that profile looks like this. So it's pretty similar. But what's special about this approximation is that it's a linear combination of 2 out of the 10 sort of columns in the dictionary matrix. So the first column captures the effect of homologous recombination that we've learned automatically from the data. And the second captures the effect of end joining, which we've learned automatically from the data. And what we get is a representation of H2AFX dependency or fitness as a linear combination of these 2 sort of pathway elements added together. And so the mixing weights or the loadings are represented here. So, there's slightly more HR than N joining. You can see that these two pathways that we learned automatically sort of have different profiles that are like different drugs will induce dependency and N joining versus homologous recombination. But when you add those two profiles up, you kind of get what looks like H2A effects. So if you remember from the introduction of the talk, this is exactly the kind of approach that our problem that our approach was trying to solve. So H2A effects is a known pleiotropic gene. Again, it can activate HR or N joining depending on the toxin. And so what our model has done is it's learned representations of these two pathways automatically from the patterns in the data. And then it uses those representations and composes them to sort of model the effect of H2A effects. And is basically modeling the fact that different pathways can be activated in different toxins. And that gets you sort of your representation of your single gene. So I'll sort of end with a larger example, zooming out to a much larger data set. And this is called dependency map. And so this is a very notable data set that Broad. Lots of collaborations have been started from mining this dependency map data set. But the basic setup is very similar to the original screen, just scaled up a lot. So rather than taking a cell line and treating it with chemicals, you're actually just taking diverse cancer cell lines. So cancer cell lines from all over the country, different patient samples, different tumor origins, metastases, primary samples, et cetera, that can grow on a dish. And these have all been screened with the same genome-wide CRISPR-Cas9 library. And so the result of this is really this rich resource of which genes are responsible for cell growth in which cancer context, which is a great starting point for our model. And so we applied Webster to this data set, and we recovered a sort of a dictionary of 220 biological functions that encompass dependent fitness patterns seen across 675 cell lines. And then we mapped each of 3,000 genes onto up to 4 of those functions. So it's just kind of a scaled up version of the analysis that we performed for the genotoxins data set. I'll just give you one example of the kind of representations that we get from applying it to a larger data set. So SHOC2 is a notable cancer protein. This protein is sort of a signaling factor that's at the heart of multiple different upstream signals. And so different combinations of activated signals can converge on SHOC2. And so lots of different cell lines will depend on SHOC2, but for slightly different reasons, because they have different kinds of activated signaling pathways. So this is the measured fitness profile in dependency map. So again, SHOC2 was knocked out over 675 cancer cell lines. Cell lines in purple are more dependent on SHOC2 for growth, so they have a higher fitness effect when you knock out SHOC2. And our model approximates this profile, but as a combination, once again, of 4 out of the 220 learned functions that are learned automatically from the data. And so similarly before, when you map these onto biological priors, you can kind of recover sort of the meaning of these different latent factors or functions. So they represent 4 different signaling pathways. And each one of those pathways converges onto SHOC2. So it sort of recovers, again, this pleiotropy concept, where in different kinds of cancer cell lines, different sort of pathways are activated. But if all of those require SHOC2 for function, then composing them together linearly recovers the original SHOC2 measurement in a way that enhances the interpretability of why SHOC2 is actually important in those particular cancer cells. So one last thing that this kind of analysis enables is really exploration, right? So this is an unsupervised method. Again, these kinds of labels, these highly interpretable pathways and things, are only being added after the model has been performed. So it's really a numerical analysis from the start. But because of that, it allows you to sort of explore the data in this sort of unsupervised way. So you can imagine taking, let's say in this case, all of the genes that have fitness effects in DEPMAP and all of the functions that we've learned automatically and kind of project them into an embedding space that we use to visualize our results. And so in this particular case, we're starting with 3,000 gene effects and 220 functions. And then we can plot them sort of like this. So What does that look like on real data? So this is sort of that embedding space with genes only. Again, each gene is embedded into this space depending on which cancer cell lines they sort of kill when they're knocked out. And you can kind of see like a very clustered structure to this embedding space. And when you annotate on with our learned functions, you can see that these functions kind of exist within or at the center of different sort of co-essential genes. And that's exactly what our method is trying to do. It captures the effect of these co-essential genes and it sort of learns a representation of that module, and then it keeps it in that dictionary. So this is exactly what our method is designed to do. And again, when you start to annotate these functions, you get these interpretable bioprocesses. This is a really fun map to explore. And Andrew Tang and others at the pattern team at the Broad have built an interactive portal for this. So if you follow this URL, you can sort of mouse over these data points and get a lot out of exploring this data set that way. And finally, you can even start painting on sort of even larger sort of annotations. For example, mitochondria, lots of different functions happen in the mitochondria. We have annotations for which proteins sort of are involved in those processes. And so we sort of recover like certain interpretable compartments in the cell based on these annotations. So that's really fun to explore. So that's the end of my sort of whirlwind tour of the method. There are other things you can do besides what we've talked about, and I will refer you to the published paper for more detailed examples. One example is, for instance, taking compound screens and aligning them to genetic effects that you've learned from CRISPR data. So compound screens are inherently very noisy, but they can be applied over panels of hundreds of cancer cell lines very readily nowadays due to PRISM. And so you can take a PRISM profile and model it in terms of these biological functions that you learned automatically from CRISPR data. But to conclude this part of the talk, we believe that this kind of unsupervised learning of gene pleiotropy is pretty tractable. We recommend starting from a data set where you have these gene perturbation effect measurements. In our case, we analyzed fitness, but there's no reason that this doesn't work for other kinds of phenotypic measures as well. And in order to accomplish these sort of interpretable latent variables that you get, we recommend using sparse approximation embeddings for this task. So I'm going to stop my talk here for the sake of time. And I have a few bonus slides after Marinka's presentation to expand on some of these themes as well. But for now, I'll skip to our acknowledgements slide. Again, I'd like to acknowledge my postdoc mentor, Bill Hahn, as well as amazing collaborations with Marinka, who's on the call, as well as members of the cancer data science team. And so with that, I'll pause maybe for any questions that have arisen, and if not, pass it over to Marinka.
Thank you so much, Josh. There's one question in the Q&A. Thanks for the great talk. How could you determine optimal parameter T, being the maximal number of genes, which constrains the complexity of functional representation? I think it depends on what biological question you want to solve using this method.
Absolutely. So one of the key hyperparameters is T, which is, again, for those who are interested, the number of sort of latent variables or functions that you use to approximate your gene effect. And yeah, we determined that sort of empirically using a sweep. So we tried different values of T as well as K, and then swept over K being the number of latent variables or functions. And we looked at metrics like our the loss or different cluster purity metrics based on gene sets and things like that. We have a NeurIPS sort of workshop version of our paper that goes a lot more into those methodological details. So I will actually refer you to that particular version of our manuscript. But yeah, it's really simple. We just swept over hyperparameters. Those hyperparameters will change based on which data set you're using and how big that data set, how complex it is, et cetera. But that's a great question.
I just encourage anyone to raise their hand or type in the Q&A, but maybe we'll go on to Marinka now.
Thank you, Josh, for this nice overview of the Webster approach and the insights it has enabled. In the next few minutes, I'd like to zoom out a bit and speak about broader implications and impacts and opportunities that representation learning paradigm creates to design high-capacity AI models to predict other traits and phenotypes. So the algorithm that Josh presented and Webster would very broadly fall within the realm of representation learning. And representation learning is a very exciting research area in machine learning that really has fueled advances that we have seen in AI over the last few years. And at the very high level, the key idea is that we start with a complex input data set. And that data set might often span multiple biological scales of organization. It might contain different heterogeneous multimodal readouts. And with such a complex data set, we use a representation learning algorithm to extract what are essential informative and predictive features or representations that one can then use to derive predictions, create and extract patterns, and provide insights. And so the way this is done is through representation learning algorithms that are essentially optimizing multiple layers of some nonlinear graph-based transformation. And I will give an intuition of those algorithms on this call very briefly. So if you think about your favorite machine learning application or biological question that you wanted to tackle using machine learning, the way that to think about tackling the problem is typically to go through a particular kind of life cycle or a series of steps to arrive at a high capacity, accurate predictive model. And so those steps are typically that we start with a data set, and we want to extract from that data set that might be really kind of various and heterogeneous in many ways. We extract structured representations or feature vectors or a list of variables or covariates or embeddings. Those are all the kind of the names that we are typically using to refer to this concept. And those structured representations are then used as input to a machine learning predictor of your choice that might depend on a particular application that you have in mind, and that predictor is trained to derive predictions and insights. We often refer to the second stage of building a predictor on learned representations and training that predictor as downstream prediction task. So traditionally, the way we went from the input data set to structured representations is through the process of manual feature engineering, meaning that a designer of the algorithm or the one who was working on application had to think very hard about how to represent each unit of analysis, whether that is a patient, a gene, a function, with a small number of informative features that can be then used to train a machine learning predictor. So what representation learning allows us to do, it allows us to automatically learn those essential features and those structured representations without the need for human experts to manually feature engineer those descriptors, those essential features. Okay, so how can we do that? So in essence, the way to think of representation learning is the following. So we have on the left an input data set, which I'm visualizing as a network or a graph. So meaning that the data set is represented as a set of nodes connected by lines that indicate associations or edges. Think of that network as a gene function relationship network or a gene-gene interaction network, protein interaction network, cancer dependency data set. Representation learning algorithm is a function f that will take each and every node from that network, for example, U, and it will map it into some d-dimensional embedding space and represent that node U with a vector of features. And that is particularly important because once we have these vector embeddings, then we can use the power of statistical methods, decades of machine learning techniques and predictors that were built that operate on those vectors to build different kinds of predictors in downstream tasks. So the particular kind of functions that allow us to go from the input data to this low dimensional embedding space that are powering recent progress of machine learning in biology and medicine often include functions that operate on this graph structure data and are called graph neural networks. The way they operate, and that is also true for Webster, is intuitively they do the following. Now, if we have in our input, we have two objects, say node U and V. Think of two genes, U and V. That function, representation learning algorithm F, will take each of those two nodes. It will represent them as points in the embedding space, and it will do so in such a way that 2 points U and V that are considered to have similar local topology in the input embedding space, they will be embedded as nearby points in the embedding space, meaning that the embedding space is optimized to reflect the topology of the input graph. 2 points similar in the input data set get represented by points that are nearby, close to each other in the embedding space. And once that embedding space is built by the algorithm, then it can be used to train various downstream prediction tasks. So the broader implications is now that we have powerful representation learning algorithms developed, and there are many architectures that can learn automatically those representations, we can apply deep learning much more broadly. So beyond just medical images that where we have already seen tremendous progress that architectures such as convolutional neural networks, recurrent neural networks, and recent transformers have brought on, and on biological sequences, we can now apply deep learning and representation learning on any kind of structured, complex data set. So this creates new opportunities in biology and medicine. One opportunity of that type was mentioned by, was discussed by Josh in Webster. And there are many other kinds of applications across biological, knowledge, therapeutic domains that really use the power of representation learning to model molecular graphs, protein structure, ontologies of cellular systems, knowledge graphs, gene interaction networks, single cell networks. So in the remaining 2 or 3 minutes, I'll just give you a very high level overview of a few of those applications. So with representation learning algorithm, what we can do is we can learn those representations that are context dependent, something that also was alluded to in Josh's talk. What would that mean? We can learn a representation of an object such as a gene that would vary depending on the biological context of that gene. It would vary depending on the cell type identity. It can vary depending on patient identity. Once we have those cell type patient specific representations, one can use them for various downstream tasks. For example, here I'm just showing 2 examples of how cell type specific and patient specific protein representations can be used to design then ML models or that can predict therapeutic action and disentangle it at the single cell level, or build predictors of patient response to various immunotherapy treatments. In a related problem, then these representations can be used to understand how or what are possible targets for a novel drug in development, and what kind of therapeutic indications that the drug might be represent a good opportunity for. And if with that application in mind, one would learn representations on a larger heterogeneous structured data set, which we would refer to as a knowledge graph, and then learn representations of genes and drugs in development and diseases on that knowledge graph, and then use those learned representations to predict and nominate what might be promising therapeutic opportunities for a novel drug in development. And doing that with representations can provide substantial performance gain, especially for hard to model settings, such as diseases with few treatment options that have limited molecular understanding, because extraction of essential features by representation learning algorithm is done automatically, and it's optimized to produce strong performance on downstream tasks. Finally, there are other kinds of biological problems that representation learning can be particularly useful for. We have seen in recent year tremendous interest into using these graph-based representations to model binding affinity between therapeutic targets and novel compounds. And the way that is approached is by taking the structure of therapeutic target, use a separate embedding algorithm to derive a feature vector and embedding for that protein target. Now do the same for a novel drug in development to derive a drug or compound-based feature vector, and then in the ML predictor, take those feature vectors as input and train a classifier that can predict the binding affinity between the target and the candidate compound. And those predictions can be then used to prioritize therapeutic targets based on the likelihood of binding to the desired effect. There are ways for those representations to be explained, which is a really important point, as they are embeddings and representations are automatically derived by algorithms, and as such they often are perceived as black boxes that are very hard to interpret meaningfully. But with recent algorithms in explainable AI, such as GNN explainers and others, it's really possible for domain experts and users of these models to ask queries, ask questions, and get as output not only point-wise estimates or predictions, but also information about what part of the input data has driven the ML predictor to derive a particular embedding and make a certain prediction. And that can be used, that information can be used to decide when to trust a prediction and what prediction is most most trustworthy to be used for downstream experimentation in the lab. So let me conclude by just highlighting some of the resources that are available to you for these kind of problems. And I will mention one open science initiative that we have started at Harvard that is called Therapeutics Data Commons. You can learn more about it if you go to the website tdcommons.ai, but it's meant to serve as an ecosystem or a meeting point between AI machine learning scientists who are designing methods and domain experts who really identify what are meaningful tasks, data sets, and problems in order to facilitate algorithmic and scientific advances. And so most importantly, the way you can use TDC is to really obtain support throughout the life cycle of machine learning models that we have seen at the beginning of my presentation. TDC can help you to retrieve, process, and harmonize data set to transform it into an AI-ready data set. Then it supports interaction with variety of libraries that are today's most popular algorithms for machine learning models, including PyTorch, TensorFlow, JAX, Hugging Face, and others. And then once you have a model, TDC has functions, visualizations, and evaluators that can evaluate the generalizability, accuracy, and performance of the model, and compare the model to others for the same application in order to identify what is the best-in-class model to be transitioned for real-world application. And the way this is done is by this rich programming as well as interface and a Python library that TDC provides to support researchers throughout machine learning lifecycle. So I will stop here to still leave a few minutes for questions, and happy to take any question or open this for discussion. And I believe Josh also has a few concluding remarks and some discussion points to share.
Yeah, that was a great, really rewarding high-level overview of the representation learning field, especially applied to biomedicine via graphs. I think this is a good time to open it up to questions from the chat. And then, yes, I do have bonus slides in case there aren't questions. But yeah, let's definitely get into what people are curious about learning from today. We have plenty of time for that.
Thank you both so much. One thing I was curious about is if you thought, you know, many times when you're taking a new therapeutic to market, a lot of the first stages are assessing safety and looking for off-target effects. Have you thought about using your same paradigm to predict what kind of specific toxicities or side effects you may anticipate with your compounds?
Yeah, that's a great point. We have, in my lab, we have a few projects where we are looking at modeling cardiotoxicity as well as liver toxicity with a focus on novel drugs in development. I think there are a lot of published methodologies and papers that kind of tackled safety and toxicity problems from various perspectives. Major limitation that I see in the literature is really being aware of what kind of data is available at a particular stage in discovery. So that means that when early on there is very little or no patient data available, so it really then becomes a question of how one can carefully and source data available at early stage of drug discovery to be able to anticipate safety events in later or clinical stages of discovery process. Josh, do you want to comment do you want to add anything, especially in relation to Webster?
No, that's great, Marinka. Yeah, some of Marinka's most famous work is in this line, so I definitely encourage you to check those out as well. Yeah, in Webster, we have a very simple view on this, where if you screen a compound, for example, through a set of cancer cell lines, we can take advantage of the representations that Webster has learned to model these compounds that have been screened, through PRISM or GDSC. And so there are open lines of inquiry down that front as well. But yeah, I refer you to the paper for more details on that.
Great. I don't see any hands raised or questions typed in the Q&A, so we'd be very appreciative of your bonus material, or I can keep asking you guys questions I have about your exceptional work.
There's sort of one bonus material that I just thought I'd highlight for this audience, which is relevant for the concept of discovering gene targets using multiple kinds of phenotypes. So for the particular Webster work we were discussing earlier, we were very interested and focused on fitness effects. Fitness effects are sort of scalar measurements of cell growth differences. And we rely on these large cohorts of cancer cell lines to give us this high dimensionality, right? So we have hundreds or now even 1,000+ cancer cell lines. How do we factor that into meaningful latent variables? But recent work out of the Weissman lab that I'm going to highlight has actually done something quite conceptually similar, but in a very different context. So they've taken genome-wide screens, but they've applied them to single cell. So instead of looking at the growth rate change in, let's say, 1,000 cell lines, you look at one cell line and you look at the transcriptomic change. So that's also a high dimensional gene perturbation effect measurement, but it's done in a very different way. And also, this is actually work coming out of the Broad via Paul Blaney's lab and Ian Cheeseman's group at MIT, have done very large scale gene perturbation effect studies, but they've perturbed cells and then looked at them optically. So this is done through optical screens. And each one gives a different sort of, I would say, lens onto what the gene perturbation effect is, right? So there are different time points, different assays, and sort of different phenotypes overall. But just one very interesting tidbit that I'll leave the audience with, and I think we can break for more questions or in the call, is like gene function discovery, right? So I didn't mention this in the talk, but as part of our work, we analyzed a particular protein that's called C7orf26. Its function at the time was unknown. And we could represent that protein as having an effect on the integrator complex, which is a known protein complex. But in particular, its strongest effect was matched to a very small module within the integrator complex that's composed of 3 of its proteins, 10, 13, and 14. And through some biochemistry in the lab done at the cancer program, we figured out that indeed this unknown protein, that's a human protein that's expressed in a lot of cells, but just don't know what it does, turns out it binds this module. And so this sort of 3 protein complex module sort of became a 4 protein module. But what's really exciting is if you dig into these other papers. So for example, I mentioned this Cell paper that's out, I think last week from the Weissman lab. They did these gene perturbation effect measurements in transcriptomic space rather than fitness space. And you can do very similar analyses, right? Clustering genes, et cetera. When they clustered their data set, they found once again that the integrator complex had this modular structure, which we could see from our fitness data, right? But they're looking at transcriptomic outcomes, right? So clustering transcriptional programs gives you this protein complex. And once again, it brings in C7orf26 as a highly specific member of this very small part of the complex. And then overall sort of gives you the complex as a whole. So this is a very interesting parallel, right? And there's no question here, it's just a discussion point. What happens when we start doing many different kinds of these screens? I think methods to analyze and think about multimodal data might become very important. And finally, just to point out the optical screen as well. As I said, they knocked out these same proteins and looked at the morphological consequences and the morphological effects. And in their paper, once again, this is Funk et al. In bioRxiv, they also found that C7orf26 is co-phenotypic with this very specific module. So this is kind of a bonus slides, but food for thought for the audience and what the future of screening holds when we start to do these more and more often.
Well, thank you both for your presentation today, for sharing with us your recent work and ways that you've been using the combination of genetics, large data, and machine learning to advance treatment. Hope you both have a lovely day.
Thank you.